Good morning, good afternoon. Thank you very much for having us here. Marwan, it's very kind of you and your team to have somebody from Pittsburgh come in and talking to you at lunchtime with these wonderful people trying to eat. So I hope that I don't show any disturbing images during your lunch. You can have, and I will try to save on the science and try and be more on the focus of what we've done. So what are we talking about today is need glasses for everything now. So we're going to talk about what we developed over these last 5 years in our lab at the McGowan Institute, where we developed something that we call the new Ex-Vivo world when we first started trying to develop something new for organ preservation, and we realized there was a little more complicated and more complex than that, and I will try to share some of this data with you. So first of all, the conflict of interest, I like this confluence of interest, was published at the JAMA a year ago, saying that have to be a conflict to have some interest in on what you're doing. So otherwise, you're not going to have an interest. And so I am a co-founder of this company that I will be talking about in the way that I'm the chairman of scientific board advisor, but I don't have any financial meaning on this company or the role on this company. So by the conflict of interest in office of my place, I'm capable to keep my career as an academic surgeon. I don't know for how long, but this is how we do it. And I want to thank the people that obviously work with us in our lab and do most of the work. There's a lot of people that you will see. They're not physicians. They are PhDs. They are bioengineers. They're perfusionists. They are lawyers. There's a lot of people working with us in different roles, and I really like this because in the end, what I discussed this morning here with your Innovation Institute, if you want to do something different, you've got to get people that think in a different way to do this properly. And I want to thank my boss now, together for 25, 26 years. Next month, he's going to be 90 years old. And when he hired me in Pittsburgh in 1991, he always had this idea that a lot of the development that has been done on the field of transplantation has been done by the surgeons going back and forth to the lab. And this is how he built his legacy in Denver before he got into Pittsburgh. And I was already doing this in my home country, so it was an easy assembling to go to see what he's been built and what he done. When I got in Pittsburgh, they were doing close to 500 transplants a year, liver-wise. And this was a real powerhouse, a real transplant enterprise. And obviously, with the time and everything else, the numbers went down. So I became director of the liver transplant program in 2001, and I realized there was a lot of things that we have to do to try to keep up and go back to our legacy or to improve how we were doing things. We could do things in a different way. So one of the things that he always showed is patient-centered cycle of medical innovation, where you have a clinical problem, and with this clinical problem, you're trying to build a model and try to work around. Initially, just with animal models, and as you know, right now it gets real more complicated. You can go to cell, you can go to transcriptomics, metabolomics, you can do even simulation, you can do in silico trials. So it becomes a much more complicated field. With this approach that I always thought about, the right methodology, some of the studies you will do in a holistic approach, sometimes you will do a reductionist approach. So you have to find the right method and the right environment to do this work. A lot of the work done in this field comes from an initial idea where you start doing experiments, and then you have to have data. And obviously, there was all the risks and benefits of this. You don't get a lot of love from the NIH to run large animal, preclinical large animal trials because it's expensive. It's not considered very good science. And then within this data, you can then go straight back and what happened to me with the new IPs and new companies and grant submissions and eventually go to the benefit side of it. So when you look at the transplant process, I don't know if all of you work with transplantation, but we were taught that it was an easy pathway going from an organ donor to an organ evaluation to procure the organs and then do transplants. So there was not this concept of working with organs ex vivo, as we're going to present today, because most organs, the blood was flushed out of the organs, and you have the organs sitting on ice for a given period of time, and then with that we call organ preservation, and we're all happy with this. We built a whole field on this. So when I came to Pittsburgh initially, I was interested being here, where did the organs come from? Because there was a lot of ways that we could get offers from someplace else. So I was telling Marwan yesterday, I was part of the group that built an organ referral center in our place where we were dealing with our own referrals from OPOs from other parts of the country. And as you can see rapidly, the number of offers that we were getting up every year went up to 12,000, ended up being up to 15,000 organs a year. So how do you manage in your daily life when you're getting offered 15,000 organs a year? In the first beginning, it was good. I was able to get to all-time UPMC record. We did 850 transplants in 2007 and got some awards for this. But it was a very thin system because when you do this amount of volume and you spend this amount of money transporting organs around, $2 million a month flying organs around, it is a thin system with a lot of issues with allocation, and I didn't like that. One of the good benefits of this, I was very close to the heart and lung people, so I was able to do a lot of things that I really enjoy. I like doing this combined heart and lung and liver transplants, and we published this and we present this in San Francisco a few years ago. We had them doing 22 combined cases where they do heart and lungs and liver at the same time, and I was very happy to work with this population that now there's some issues with the allocation that people don't like. What do we do for this? So in this transplant world, there are several ways that can impact on your process. And I choose to go in this segment here because actually I had the chance to work with the people from the business school. And I asked them very clean, what was wrong with transplantation? Why we're not going back to the way that we were? And besides all the medical issues that we have, is that you have one major problem. You don't control supplies. So your transplantation is based on your ice box of an organ sitting there. You're running against time to do a case, and everything else is going against you. So it was clear to me that preservation was a big issue on. And when you look at the data, and you probably have to say anything new, the number of brain dead people in this side of the world is going down because the ICU is getting better care. And most of the patients now that become organ donors, they have a higher age, a higher BMI, higher rate of medical problems, being diabetes and hypertension. So the so-called improvement of ICU care has an impact on the transplantation because there are less patients now that will be seen as brain dead patients. So what that did to the field, that we have a lot of increasing on the so-called the DCDs, the donor after cardiac death. So when we started doing this many years ago, the discard rate was very high, and then we go down to a place that we thought that we could be able to use everything. And then as I showed, there was a change on this population, and then you start using less organs, and the discard rate goes significantly high. And for DCDs, the discard rate is higher and higher, and what would really bother me as a clinician running my program was that we doubled the amount of patients that were invited to leave the list. And that for me is devastating because that means that you're going to die. So by the end of the day, with all the eval, financial clear and everything, you are on the list for 3, 4, 5 months, and I come back to you one night and said, now you're off the list because you were too sick. I actually hated that because it was a death sentence that I was giving to this patient. I didn't like to be part of it, even though I was doing this most of the time. So the same group in the publishing that if we keep the same rate that we use organs, we end up going to a situation that we throw away more organs than we use. And that is also embarrassing when you look at the families of the people donating organs to us, and I spent a lot of time. I did donors myself for 10 years flying around this country, and they are the true heroes of this business. So when we don't use the organs because we don't know how to do it and they're not used enough, and the discard rate is that high, that's going to be a problem. So in our own data at Pittsburgh, we have a single OPO core that does a great job. The discard rate for DCDs in livers was 50%. It was even higher than this, 50%. And this is a problem down the line if you're going to try to run a better program. And for the people that don't do organ transplantation, and this name's brain dead donor and donor after cardiac death, what means basically is this population, we control hemodynamics and oxygenation until the very end when we procure the organs. In this population, we don't. So there is a lot of warm ischemia time built in this population that can be a major issue for the organs to be used down the line, and I was concentrating on that. So it was clear that there was an equation between time and organ quality, and the longer the time that we have, the worse was the organ quality on because of the lack of oxygenation with the ice bucket. So could we create a system ex-vivo that we could move this curve up and be more effective in rescuing these organs? And obviously, is not the most bright people that actually lives through, but the one that knows how to survive the changes on. And I like this quote from Darwin because it makes an impact on my own practice. So an active transplant surgeon doing this for 20 years, after doing 1,500 liver transplants for 10 years, I decided to go back to the lab, a different lab, actually not my own lab, because I wanted to have a place where I could work with people that were not exactly like myself, who were not surgeons or physicians or nurses, but they are actually bioengineers and scientists and PhDs that will see the world in a different way. And they have a very interesting system built at the McGowan Institute going on for 10 years, where they were very good in developing medical devices. And I will talk a little bit about this, of the regulations, the legal issues, the financial issues that will be embedded on this. Like everything in life, you can't do anything without money. So as I just said, it's hard to get grants early on if you don't have good data. But I have this wonderful human being who was my patient that I transplanted her many, many years ago when her husband is an investment banker, and they want to do an endowed chair for me. And I said, it's not a good business because the money goes to the hospital. We don't see a lot of this in our lab. So they decided to give me a lump sum of money. They gave me $1 million check and said, see what you can do with this. See if you can build your system. So the idea was to go back to that equation that I showed you and see, can we have a system that actually improves the quality of the organ over time, and we get rid of this ice box and this lack of oxygen thing. And you know this well, the people that do this on the field, that the lack of oxygen and low temperature has a major impact in the way that the cell process energy on, the way that the mitochondria function, and the way that the membrane function. Then you can have significant and irreversible damage to some cells under hypothermia and anoxia that you can't recover this. So when I came into this field, I have two missions to do. The first mission was to develop the best possible device that I could away from the eyes. And the second mission, and I will show this as we go and why we did this, I want to develop a solution who would be a blood-free, a cell-free, hemoglobin oxygen carrier solution that we could oxygenate the organs very effective at any temperature. So with that mission in mind, what we did, we set up a line of work. There was a development platform. Because I'm a liver guy, I want to go to the liver first. And the next in line were the kidneys. But interesting enough, I sent a grant to the DOD a few years ago to work with the limb transplantation, which we call composite tissue allografts. And I was funded on this grant. And I ended up devoting a lot of my time to this, and now became a major part of our development now. So right now, the launching order is liver first, and then we're going to the CTAs, and now kidneys are going to be coming next. And then the other organs will be coming after that. So I will show this figure a couple of times because it's interesting when you look from the 10,000 feet that sometimes you are trying to resolve a simple problem in your life, and you are focused on an application to preserve organs, and you create more problems that you can fix around, but you learn how much you don't know or how much things could be done different if you approach the situation in a different way. So I will make some brief comments on how we develop these other things on, including this one at the end, where instead of trying to preserve an organ, I will show you with the system, we are actually trying to rebuild an organ from the very beginning with the whole scaffold and with the whole ECM. So how this machine perfusion works, actually works. So I am working on this chapter now with, on this paper with the people from Europe trying to say, how do we call this machine perfusion? Are we divided by the flow? Are we divided by the temperature? So we agreed that we are going to call this by temperature right now. So as the temperature goes, there's hypothermic with 4 degrees Celsius. There's normothermic, and you guys are engaged in 2 clinical trials now to go to normothermic. And we decided to go right between these two universes on where we want to go to this temperature, and we'll talk a little bit about this so-called temperature, the normothermic temperature. But for that, you could not use red blood cells because there's a problem with the temperature and how the cells function ex vivo. So what I did initially after going around the world, literally for 3 years, I found a group in the Netherlands that could put up with me and work in our system, meaning that I want to have the bioengineer into my lab working with us to develop what I thought was the best for our patients. And I found these guys in the Netherlands, in Groningen, and they've been working with me now for 6 years. It's a very nice device with the pulsatile pressure. This is the pump. This is the oxygenator with pulsatile pressure to the hepatic artery, with a continuous flow to the portal vein, and you can also collect bile, and the liver goes in an upside-down position. I will show this in 1 minute. How does it look? Obviously, when you go into business, you have to know who is your competition. So I met with all these people in different times of my life. They're doing a great job. They're investing, some of them investing a lot of money on this. And I have some differences in how I saw the way that they were moving this business and the medical and the scientific part of it. So I was offered to work with some of them, but I thought that the best way for what we want to do was trying to work with organ assist and build a new solution with them. So Jim was the pioneer in machine perfusion, and I tried to work with Jim. He's a great guy from Columbia, New York City. And his first data showed that the oxygenation levels at low temperatures were almost half of the way that we have with our system on. And even that, this oxygenation was very limited. The lactate was actually going up on their system over a short period of time. And what we saw on and on and on in our livers, and actually the liver clear lactate, and the lactate goes down, and the liver makes bile. So we have a very interesting initial thing about oxygenation and different pressures on. So what would be the ideal preservation solution for machine perfusion? With all these issues that we have with viscosity, osmolality, oncotic pressure, and oxygen carrier capabilities. I'm not going to bother with the biochemical details of this, but I spent a lot of time on this, beginning with the blood bank. I also trained in cell transplantation when I first came to Pittsburgh. I was doing islets and bone marrow transplant. And using blood outside of the body is a problem. It doesn't look like this clean with all these RBCs, beautiful as they show on the picture. There's a lot of issues with the RBCs itself. Then you end up having this. You have a lot of hemolysis on. So when you use the blood ex vivo, there's a lot of things written about this. You're going to have problems with this hemolysis that will have an impact on how you preserve these organs down the line. No matter what kind of pump you use for this, you're going to have problems with the same issue with hemolysis on. So one thing that was clear to me, I do believe in evolution. So 600 million years to develop hemoglobin, it was clear to me there was not perfluorocarbons or other molecules, that hemoglobin was the most effective molecule. But how can you make this work? How can you have this complex molecule to be used in a way that is capable to take oxygen? So regularly, these hemoglobin molecules within the red blood cells, they will go to the wall of the cells, and they will bring oxygen into the mitochondria. So some of the work that I will show to you, I will be focused on the mitochondria, because it was very interesting to understand how was the work inside of the powerhouse of the cell. So I went back to Dr. Belzer's work from the 60s. This guy published this on the 80s, but this is work from the 80s. When they developed the Belzer solution, they were trying to do Arrhenius curve to look at the mitochondrial function and look at the lowest point of the curve where the cell was not active at all. And I came back on the same data 30 years later to see what was the lowest, highest point that I could have some functionality of the cells in a temperature that could be ideal for us. So it's interesting in science how sometimes you go back to the literature and look at old data in a different way, and obviously you learn a lot from Dr. Belzer, who was the pioneer on this. So the UPIT model that I'm showing to you today is this between the two, the cold and the warm, when we go subnormal thermic temperatures at 21 degrees Celsius with an oxygen carrier solution. What was the problem with this? When I worked with this company, it was a bovine-driven hemoglobin oxygen carrier, where they isolate hemoglobin molecules from bovine and do a polymer out of this. There was some good advantage on this, but there was some problems with this molecule. So I ended up changing this molecule. We combined this with another solution. We changed the oncotic pressure. We add buffers. We have some different things that we add to this solution. Because in the end, what was important to us was between the risks and the benefit of these two solutions, I want to have a solution that have a very small molecule, because the RBC, as I will show you in a minute, is still a big cell. So when you go to microcirculation in DCDs, you're going to have some microthrombi for these cells that will sit on the tissue that you can't get rid of it. And for this, we got lucky that we actually patented this a few years ago, and this is why we built this. So there was a problem with this field. All the companies that work in the artificial blood HBOC system failed. They run clinical trials, they went to bankrupts, and they ended up getting out of business. And I went back to the so-called battlefield, and I decided to work with these guys first. And I thought there was something to be learned, but in a different way. So in my pathway, I have to do most of the work with this bovine solution. We did a lot of the preclinical. I did a lot of work for the FDA on the pharmacology side, and did some work with the FDA on the regulations. But I have to go back and develop the human side of this, and now try to show that they are similar. So I just told you that some vessels are so small that RBCs will be stuck in, and they will not get out of it. So you have ischemic damage downstream because you can't perfuse this. And the people who work with livers every day, this is how the biliary plexus look like. It's a very teeny tiny system now that you have to perfuse properly the microcirculation. Otherwise, you're not going to get that cells enough oxygenation on. There was a group from the NIH, and I don't make this to be funny, but now this guy is the chairman of internal medicine in my institution right now. But at that time, he was the #1 enemy of the HBOC industry. They took the industry down by showing that there was a problem with the free hemoglobin molecules going to the endothelial cells and having some problems with nitric oxide and closing vasoconstriction of these vessels, which we didn't see in our model, because in our model we dilute this and we increase the oncotic pressure, and we move away from the word that was a blood substitute towards a solution that was able to actually carry oxygen into the plasma. So how this thing works, little video for you, rated R. So this is the system that we developed with the Dutch group. This is the portal vein pump. This is the hepatic artery pump. This is the heating unit. This is inside of my lab. This is a pig liver. You can have the temperature and can have the flows. It's a single button to operate this. I'm not selling this device, by the way. The liver goes inside in an upside down position. This is the bile duct with the bile coming out. You have an arterial cannula and you have a portal vein cannula. I'm going to show you in 1 minute. The pulsatile flow, that was very interesting to have a pulsatile flow. So we're going to show a side shot of this. And the tissues look really good. And I will show some of the data that we have. We were one of the few groups that did all the way through and actually transplant this liver. This is the artery having a pulsatile. So because I am a surgeon, I am paranoid with details. I want to know what was the oxygenation inside of the tissue itself. So what we did, we got this company from Oxford in England that they developed these probes that you could put inside of the tissue. So I put these probes inside of the liver through the capsule to see, and that I could read this on this device. I'm not selling this device. It's not FDA approved either, but I use this in my lab to see what was the oxygenation. So at the baseline, when I open up the donors and I put these probes on, the PO2 inside of the tissue was around 80. Then when I cross-clamped, flushed all the blood out, put UW in, took the liver out, put on ice, did the back table, then put my solution in, put on this machine now, the PO2 was 200. So the amount of oxygen that we were delivering to the tissue with 3.5 grams of hemoglobin, it was huge at 21 degrees Celsius, to the point that when we calculated, we are giving almost 8 times more oxygen than the tissue was actually using. And that comes a problem in life, like everything. If you're doing too much, you might be doing something wrong. So I have a group that works with me, and I will show you in a minute. They actually isolate the mitochondria out of the tissue, and they study the mitochondria on this Clark's chamber. So Dr. Shruti Shiva run these assays for me and show within the mitochondrial function, when you look for ROS, for the free radicals, we are actually making less free radicals than we are doing at the cold storage. So we're not giving too much oxygen as we thought initially, even though the mitochondrial function was fine. So with that, Dr. Google ended up having us there with our patent. So this is a public domain now from 2012 that we have this patent. But the question is, what else can you do for this? And I will not go to all these things, but for me, this will be an unbelievable thing that we are doing now. Bioengineering organs, human microchips, and even doing oncology cases where you can resect the liver and you do an ex-vivo perfusion of the liver and do the operation outside and reconstruct the liver and put the liver back in, an auto transplant. And I'm also working with our US Army, with DOD on hemorrhagic shock, and these are the 2 programs that I will mention a little bit about this. So when we did all the studies for the FDA, I'm not going to bother with this graphic. This is the important data. It was a good oxygenation curve, was a low hemoglobin content, the P50 was good, but the met hemoglobin levels were very low. I don't know if you are aware of this literature. One problem that they have in humans when they use this in vivo was a high methemoglobin level from this solution that will actually cause significant problem for these patients. So we have this data, the FDA already went to this data, and they like this. So when we decide to do our large animal trial, the idea was to compare machine perfusion with cold storage. Why? Because this is exactly what the FDA asked me. I built this methodology after having a meeting with the FDA. So I went to the FDA and I proposed a clinical trial for them with the animals based on this, and this is what they told me to do. So basically was we're going to go 0, 3, 6, and 9 hours, getting enough samples with our device to look at the oxygenation on the livers, with this device that can look at the mitochondrial function. We get samples for biochemistry, for cytokines, for these ABGs from the fluid itself, from the tissue. And as you know, liver people, we like bio. We don't take it home and drink bio, but bio is a very important thing, the bioacids that the liver makes. So I was very focused in understanding what kind of bio the liver was making and what was the role of the bio on this. So we published this last year. I'm not going to give the whole paper, but just highlight on the very first interesting thing was when you do a 9-hour model like this with pigs, 2/3 of the animal will die from primary non-function because of the metabolic rate is so high. The livers, 9 hours on the pig is like 24 hours on the human. And when we compare this with our machine perfusion, survival was 100%. But besides being 100%, the animals look almost too healthy that the vet said, you didn't do the transplant, did you? This was a sham operation, because the animals look unbelievably healthy. So for me as a surgeon, it was amazing to see that reperfusion, once you open up the clamps, there's no change in any hemodynamics in a liver like this. And so I was really impressed to see this for the first time. We also look at pathology. We have a powerhouse with Jake Dimitrios at Pittsburgh. So we were able to build the index to see ischemia reperfusion injuries, and it was clear to see significantly that 5 days after transplant, the liver that received oxygenation recovered really well. When you look at this from the machine perfusion with the centrilobular damage, the ones that did not and have enough oxygenation on. Because I was interested in understand the mechanistic effects of this, we spent time looking at cytokines. I'm probably not going to be able to see this well, but you know the big problem with the TNF, with the interferon alpha, with the IL-1 beta. So when you compare these two groups, black is the machine perfusion system. Our cytokine-mediated profile showing this inflammatory pathway was much lower on the animals that received proper oxygenation on. And the biopsy ended up showing this, but it was interesting to see the details of this. So going back to the mitochondria, as a person working with me that's able to photograph the mitochondria in the tissues very well, so we could not only see the function of the mitochondria, but we could see the anatomical features of the mitochondria. So when they don't have enough oxygenation at the reperfusion, they will blow up against the ones that look intact when you provide oxygenation to it. And this lady that I told you does this work for me, that she isolates the mitochondria from the cells so we can look at the citric acid cycle and look at the function of the mitochondria. And over a 12-hour period in a different set of experiments, we can see the so-called RCR, which is give a rate of the mitochondrial function. It was close to normal. The ATP formation was good, and the amount of free radicals, the ROS, was actually very low. So the livers on this system, they clear lactate, they make bile, they regulate their own pH, they produce urea, they secrete amino acid, and I will show some of this stuff. They will clean up the solution as much as they can. They consume oxygen, they produce CO2. So I wasn't happy with this, surprise, because as a surgeon, the livers were working out, but as a scientist, I was not showing mechanistically why was this working so well. So I spent another year and a piece of my budget looking at AFI chips. I did a lot of microarrays, looking at 25,000 genes to see what was happening when you oxygenate the tissue over this period of time. What happened with the cytokine cascade, and I show some of the data. We did Luminex in 20 different cytokines, and we worked in different ways. And this, for me, was the most interesting thing, Metabolome. So I have a company that I work with in Raleigh, Metabolome. I'm not selling anything for them. That they did an assay for me, and they were able to look at 300 molecules in the bio and 300 molecules at the perfusate that had a role in these two different groups. So now you have a much more granular way to look at this tissue. So genetics, when you look, you can look at very different ways. There are many different softwares that you do. You can compare the animals at the time of necropsy and see that the ones that did not get oxygen have regulation of the genes associated with liver damage. The ones that did not have oxygen, the ones that have oxygen did not have this. You can look at some of the markers that you will be expecting to see after you're doing a liver transplant. And these markers are very present when you see models of liver regeneration and growth. So the guy who worked with me on this, called George Michaelopoulos, is a little older than me. He's one of the kings of liver biology in the world. For him, our model looked like his 3/4 hepatectomy in rat, that the liver left in the 1/4 of the rat that will grow back again. The gene assays that we have look like this. So I was shocked to understand that a brief period of oxygenation has this impact on the genetic regulation of the organs. So to summarize this, if you flush the liver to the hepatic artery to the portal vein with this solution, by the time that we did this, with the temperatures that we did, you will improve the way that the liver actually grows, which is a very important thing for us. We want to see livers growing and the tissue being viable. But you will minimize the pathways that you see from TNF-alpha and interferon gamma that will cause liver damage. You will have a very interesting milieu of molecules being produced, and I will come back to this in a minute because we're going to talk about hibernation and how this thing relates to this. And the bio. So why the bio is so important? Because when you look at the bio post-transplant, if you don't provide oxygenation, the bio tends to be hydrophobic, which is a toxic bio that auto-digests the cells within the bile duct system. And when you provide oxygenation, you have a changing on the bio, that the bio is more hydrophilic and less damaging to the tissue, so it's actually a good bio. And with this initial work that we published, we went back to the first human case. So we wrote a protocol to the OPO, said, let us do 10 livers, human livers, discarded livers, in the same way that we did the animals, but we're not going to transplant the livers. Even if we like them a lot, by the time that we finish, the consent is for research only. So as you've probably seen, and I hate to show this, but it's reality, a young person, 38 years old, who becomes a DCD, the OSAT, once you extubate this patient in the OR, in 5 minutes goes to the floor. The mean arterial pressure, even this person has no OSATs. Look at that. It goes up and stay high for a while, and then goes down again. And the heart rate drops, and the respiratory rate is gone because this patient is extubated and is not breathing on. But this time that you're not pronounced dead, you mean you're not clinically dead, you're not legally dead because you have a heartbeat. There's a lot of damage going to your organs. And this is what we face every day by the OPOs. So what happened with these livers? Because of the oncotic pressure, I was always afraid of this. I didn't want to perfuse an organ that would turn into a basketball because you're giving too much fluid on. So the livers actually lose a little bit of weight, less than 10% of their weight because of the fluid. They keep their pH well. Excuse me. They actually drop the lactate. They increase the flow because they get vasodilated as we go. So there's a major vasodilation that almost double the flow on the hepatic artery and the portal vein. But you can't get too much flow for livers. It's a very important learning in this ex-vivo world that you can't give too much flow because you will damage the endothelial cells. The peak of CO2 is actually low. When I look at all the systems that I review a lot of papers on this field, the peak CO2 of other systems are much higher than this, and they don't come down as we do. And the PO2 pressure in the fluid was actually very, very high. So this is an ongoing project we have with our OPO now. The first liver, you probably do the same in pathology. You will cut with the Lipshire knife and go to the liver sections to see how the liver looks. And the pathology were actually happy to see that after 12 hours, we didn't cause any damage to the livers, and we're still having ongoing data from this. So what is the summary of the liver experience that we have at this point? It's found to be safe and effective for liver preservation. The animals that received this treatment did really well with less inflammation, with less ischemia, reperfusion injury. The bio looked a little bit better, and we are able to have recovery within 12 hours. And for the first time, we were looking at something on that looked like a pathway of hibernation. I will show this in 1 minute. So going back to that graphic, we thought that we were doing okay with this. And then with the studies that we've done, and I show some of them, we unravel this ex-vivo world in a sense that with this omics data that we can have, from all these things that I just showed you here, if you combine this system with system biology, with computational biology, and you change the way that you analyze your data, the amount of data that you generate and you analyze is actually huge. So it really changed the way that I was looking outside. It's easy to show like this. So if you can combine all this data with the technology that we have now to do PCAs, to do DBNs, and to do DNAs, which is different analysis that you can look at network profiles, you can look at the organs in a completely different way that we're not used to. So one little example, this is published. What were the top molecules involved in our machine perfusion group? Ribulose and ribose were the top ones of this. And when you look at the meaning of these molecules, the ribose is very into the DNA synthesis of these cells that we saw. There was the same data from the microarray. So the data was actually linking now in a very interesting way. But I wasn't happy, surprised, because when I did immunology, I never believed in a single cytokine in a single cell event. I want to see what's going on with the network. So these guys were able to say, listen, we can actually say, we can show you how these things crosstalk and how these cytokines interact side by side over time. So we can give you a very accurate information of your cytokine network. I said, how does it look? So for example, IL-18, which is an important cytokine on the machine, on the group that did not receive oxygenate, has a positive impact on IL-1 and on the other IL-1 cytokines. And with time, end up having an impact on AST and ALT. So for the guys who work with me on this, there was a very busy network of damages happening here. And when you provide oxygenation, this initial interaction was very loose, and there was a down regulation of this. So we were down regulating this cytokine. And as we go down the line, it's easy to show on this graphic, which might be a little too much, but the Kupffer cells and the hepatocytes, they both produce cytokines. And when you look at this profile in this system that they developed for us, you see the role of IL-18 on the NK cells and on the CD4 going to interferon gamma. But if you provide oxygenation, the hepatocytes produce this IL-1RA, has a feedback regulation that inhibits this function and actually minimizes this damage on the liver itself. So when we look at this data, it becomes clear to us that we can look at data in a different way. And this is what we sent to the ATC. I'm not going to read this, but this is what we sent to the ATC as far as an abstract to talk about this. Then I am sorry, it's lunchtime, the Krebs cycle. Do I like the Krebs cycle? I hate it. But when we look at the Krebs cycle, there were some very important features that we were learning on that were happening that we're not paying attention. What I mean by this, when you look at our two groups, so the green is the cold storage and the purple is the oxygen group, there was a high level of alpha-ketoglutarate and a very low level of succinate and a high level of succinate on these two groups, which I didn't pay attention because this is not what I do for a living until I showed this data to this guy who's my resident. He's an MD, PhD, as a biochemist, and wrote this paper in 2011. And he was laughing because he's saying, you are actually having an impact on the genes of your liver. So there's an epigenetic effect on these molecules, on the genes. They're not paying attention to this. And this was always new because it was related to the Krebs cycle. So with that, we sent this abstract showing the role of succinate in this pathway. And then people ask me, can you actually hibernate organs? And I don't know if you read this literature, but there's a lot of literature on this topic, basically trying to show that you can preserve humans to donate organs. It's kind of a little awkward to me. But we understand that when the animals hibernate, there will be some changes on the way that they go through this cycle that are very interesting to see if we could use this for organ preservation. So this was always the holy grail for preservation. So there are some genes that are involved on this. There are some transcription factors involved on this. And interesting enough, the two most important organs are the liver and the brain that are involved on this. So what we what we learned, without giving too much details because it's lunchtime, but the dynamics of this process involves this increase of activity in some of the transcription factors that you have that are secreted. There will be some hibernation proteins that we can actually find this into the blood and into these solutions, and I have some data on this. And there was some show of DNA methylation that goes down the line, and there will be some secretion of messenger that goes into this process. So I like this paper done by this guy in Alaska, which is a little creepy because he went to a place and he got these livers out of these bears out of hibernation. Took them out of the den and then brought to the lab, did full hemodynamics on these animals, and then sacrificed the animals and look at the livers. Not a very nice thing, but the lessons are clear. There was an interesting dichotomy on the genes that impact on the energy metabolism of the liver, the way that they handle amino acids and lipids and bile, the way that we were seeing in our animals. How do I know this? Because when I was reading this literature on the bear, the first thing that caught my attention was the oxygen consumption of the liver of a bear at 32 degrees Celsius, 34 degrees Celsius, was very close with what we saw in our animals. So they have an oxygen consumption which is very close. Then I asked my fellows in my group to break down into all the pathways that we saw in two groups, what the black bear did and what the pig did, and who were the genes involved on this. And they almost like overlap. So it is a very interesting thing because as we went down the line, I don't have this because it's going to save you a bit. When we look at the genes, we and other mammals, we have the same genes that do not get expressed because we don't get exposed to this kind of a temperature. But if you create a system that can create that drop down on your temperature, you can regulate and also have some of these genes, as I see here, some of these genes that we have in our pigs. So you have some genomic data that we are working on this right now as we talk, and I got some of this data here. So it looks like on the hibernation side that metabolic delivery is behaving like that. Can we make this last longer? Can we make an impact on this for organ preservation? So this is what I just said about the data, that you see the gene regulation of this. You see who goes up and who goes down. And a very important feature on the beta oxidation of the fatty acids, which is a key thing for hibernation, and we saw in our model. So going back to this concept of the ex-vivo world that I was talking about early on. So we could get data from our donors to look at a lot of the diseases that they have before they become organ donors. We can look at the histology and the biochemistry of these tissues, and we can look at the way that we perfuse these organs. But once we get these organs under the system, there's a complete network of new data that we can get, and I think we own this to the donor families, because if we can't transplant these organs, we should be able to learn enough from these organs to use this data to help the people that are actually healthy today as a predictive factor of medical disease that we don't know. And one of my main issues with this is fat liver disease. So I'm not going to bother with this, but we end up doing the same analysis going to the amino acid profile, showing there was an impact on the amino acid profile, on the lipid profile, that we were able to decrease the lipid peroxidation by giving oxygenation to. And on the bio, we already talked about bio. If you provide oxygenation, the liver provides a hydrophilic bio that is less damaging to the liver tissues on. So going back to our system, there's one thing that's not showing up here, sorry. Going back to our system here, at that time that I was doing this, I was invited to write a grant to the DOD showing, can you do the same thing with the limb? I said, well, why don't we build a model for this? So to build a model, there's a picture missing here. I got this grant in 2013, and I decided to do is getting the rectus muscle of these animals and implant them on the neck. You have to do them under the microscope because the vessels are very small. You will not believe when I complain about size of the vessels, the size of the pedicle of these rectus muscles, artery and vein are very small, so I got to do it under the microscope. So we would transplant this into another animal after perfusion of 14 hours or preserving on ice for 14 hours. And we presented this in Europe last year. So I have to develop a new device for this because I could not use a device that has a liver that has portal flow. There's no portal flow in this, so we have to change all the pumps and softwares and the pressures and everything around to make a new device for this. And what I did, because we had so much work done with metabolome before, we expand our work now to look at metabolomics from the muscle tissue. I was interested also in athletes. Can we find guidelines of tissue damage in muscle that do the lack of oxygen, not for the transplant population, but for people that are actually athletes, and look at the lipids. So the data is pretty much clean. We were perfusing this with the low pressures, with the low flows, with good oxygenation. It was the same thing that we saw with the livers, the very stable pH without any change. The lactate, because there's no liver here, there's muscle ones, so the lactate goes a little bit up, but not much. And then the fun really come through. When we sampled the tissues at at baseline 7 and 14 hours, this is too big, but I'm not should be able to see this. This is the liver, this is the muscle getting oxygen, this is the muscle that is not getting oxygen. Within a few hours, you see these contraction bands, which is a lesion that you see in people with MI. If you have a person with low flow to the myocardial muscle on, and you biopsy, we don't do this often, thankfully, but if you biopsy it early on, you will see this contraction band. So when our pathologists look at this, say, wait a minute, within hours, you already see these contraction bands? So once we transplant the grafts, and when 2 and 7 days, interesting enough, the ones that got oxygenate, the grafts look nice and pink. This is where we do all these biopsies on them with the sewing this. And this is when we turn the graft upside down to see if the tissue and it's all viable. And this is without oxygen. We saw some significant damage early on, and within 7 days, there's big ulcers on from ischemic damage. This is the tissue that people are supposed to transplant, like a hand transplant. So when you look at the myoglobin of this tissue that you see, is a significant amount of myoglobin, and when you see on day 2, you see this contraction bands, which is realistic what I just told you that happened when you have a lack of oxygenation within the muscle tissue one. And interesting enough, when you do the omics of this, you see ribose, again, that I show you that graphic on early on, that the livers have ribose as one of the key molecules for the difference between these two groups. And the same thing happened with muscle. And when we do the same way with the histology to do a score to see which tissue get more lesions on, again, the tissue that get oxygenation get a much less lesion after a period of time. But when you look at the microscope, it's shocking because when you look at the machine perfusion group on day 4 and on day 7, the muscle looks pretty much similar. And then there's a lot of infiltration and necrosis and even macrocyte infiltration on day 7 on this tissue. So when we present this, it was clear to us there was a big difference between giving and not giving oxygen to tissues if you perfuse them for 14 hours. The biggest issue with hand is not only the muscle, but the nerves. Because in organ transplantation, we don't transplant the nerves. We call ourselves big boys with our loops. We sew the vessels. We don't sew the nerves. And when you were doing limb transplantation, then you got to put the microscope on and sew the nerves on because you need to have functionality on your face and your hand. And I was intrigued, are we doing enough for these people to have functional hands? That really bothered me. They have a hand that would not work. So this is the machine that we developed with the Dutch guys for the new era. And we're going to do this work. We are doing this work. I'm going to San Antonio this week to do this work. I'm sorry for the images. We are doing the same thing that we did with animals. And now we're going to get some discarded limbs and perfuse. And I am sorry for the images. I am going to perfuse some of the limbs to understand, as we did with the liver, what happens with the human limb when we perfuse them for an extended period of time regarding this. So the kidney goes quick because I'm not going to show any data, but just the concept, it is a major problem. We don't know how to handle. 5,000 people die on the kidney transplant list, and the waiting time double over the next decade. So we're not doing a great job. The first group that came with is our friends, Rutger Plug, was from the Netherlands, and now he's in England. They developed and patented this system, and now they did not patent it. They were the one doing the trial. But this machine did not use oxygen. And when they look at their own data 5 years later, they were frustrated to see that there was not a significant impact on the function of these kidneys 5 years later because they just flushed them without giving good oxygenation on just by cold storage. And this machine sits in a lot of the OPOs around the country, and people make decisions on allocation of kidneys based on the profile of these machines, which for me is an amazing thing. So and when we're looking at all the papers with the meta-analysis of these issues, you can see that when people look at these different trials, it didn't seem like there was a big impact in not giving oxygenation early on. So when we went to this field, we thought that this evolution, it will help us to use this with our system. And we are doing the same thing now. I'm not going to show you the data now, but we have a different system which it looks around the same, where we have a oxygen carrier solution for kidneys to be preserved. Those are some of the friends that we've been working with. They don't face this. So by the end of the day, everything that we do here, there's a little mountain to be climbed there. Sometimes you don't survive going up because we have to go to the FDA. It's not as bad as a drug is, but realistically, what I learned from the FDA is they know there are 7 devices and 7 solutions being worked up. They don't want to hear this. When you go to the FDA to actually approve something, they want to have a single combination, device and solution, to be approved. So once you have this thing developed, you have to have the same solution all the time and the same device all the time for the next patient. So you cannot go and change all these things around where some people would like to do it. So in the end, in order to go to a clinical trial, and we're going to this right now, in our own pathway, we have to develop a new device and a new solution. Those were the patents. They have to have a license agreement with this company. We published this data. There was some companies that came out of this. The company that holds this patent, they have a contract research agreement, corporate research agreement with Pitt to pay for this trial. And I hope that we're going to get to the FDA over the next months to run our first clinical trial with the 10 patients. But what's up with this ex-vivo work? You're just talking about these things, and it became more complicated because now you are talking about ways to minimize damage or do some technology outside of the liver. So I came with this idea of having an organized CU, looking like a dialysis unit, where you could manage the organs outside of the body and try to improve their function and determine when you could use the organs. And this has a big impact on the organ transplantation work because we would like to have, at the end, improved outcomes. So healthcare comes down to results. If you want to save money in healthcare, you have to improve the quality of your results and have the patient with better outcomes. So can we get to that point by achieving all these milestones that the system could provide to us? And then while I am doing this, they are asking me, what is the future? What do you think that organ preservation will go through? So I said, initially, I am comfortable with the fact that we can rescue organs that are not using well. And if we discard this for the biomarkers, it's fine. But can we use a system to rebuild an organ? Can we turn this upside down and now use the machine perfusion system to decell an organ, take all the cells out, have the scaffold with the ECM intact, and now reseed that with the patient cells? So you're not selling a used car, you're building a car for your patient with their own cells. Can you do that? So they asked me to come and look at this NIH proposal from my own institution, the Harvard, Yale, and Pittsburgh. And obviously when they call me as a consultant, you should be nice to the people. I said, well, this is not going to work. So what do you mean it's not going to work? Well, you can decell the organs, but you don't have the connections. And in organ transplantation, if you don't have something we can connect and sew to the patient, there's no way to use these organs. So I'm going to show this briefly. We decell this liver. So this is a normal liver of a pig with all the tissue on. And when we take all the cells off, the liver looks the same without the cells. It's almost like an empty house. And when you look at the pathology of this, you see the portal trias, you see the bile duct, you see the hepatic artery, but a complete naked scaffold without any cells in. So how do you deal with this? So I'm going to go to a quick movie for the last minutes so you wake up. So I said, why don't we use the machine in the other way around? Can we have a liver into this machine right now, perfused into the portal vein, which is very toxic solution with these detergents that we can remove all the cells out and keep the ECM intact. And this goes on and on for a period of time. For the sake of time, I will move a little bit here. So I have to change all the pumps to make a pump that was more, and then this is how the liver looks. So I don't know how many times have you seen this in your life. A liver without cells looks like a ghost organ. There are no cells inside, but the organ is perfectly fine. It's intact. The architecture, the capsule, the texture, everything looks normal. The vessels are not good because the guys who procured this liver were not transplant surgeons, so the vessels were not exactly what we want. But you can do this with other organs. You can actually have a D-cell scaffold that my CEO called this farming, and I hate this, farming organs. But you can have organs like this. So how do they actually look like? If you go back, and this is a rat model, and try to see how the vasculature actually looks like. And it is amazingly that the vasculature looks completely intact. So when you infuse a dye back into the liver, this is a liver from a rat, the vasculature is intact. And we have figures like this and movies like this with arterial and portal vein flow showing that the vessels are all intact. So you have a tissue sitting outside of the body now that you could seed with cells. You find a way to use your device now as a bioreactor for this. And this is the idea of having this diagram here, is that you will decell with your machine, and then you will change the settings, and you reseed the cells now as a bioreactor. And for this, I will leave this question open because we thought that we'll be able to do organ preservation as the biggest thing in our life. And then we probably have to do a little bit more than this. I was told that my time is over, so this is about business. I'm not going to talk about business. I want to just thank you for your time, and I'll be happy to answer questions about this. Thank you. Sir.
You mentioned the 7-hour and 14-hour mark when you were using the preservation machine and the difference in the effect. How long does that effect last? Because you obviously want to last in the recipient. How do we know it's not a transient effect, or is that why the clinical protocol exists?
I didn't get your question about the 7 hours. What I did on the pig was a 5-day follow-up. It was not 7 hours. So the animals were followed up for 5 days, and they have normal liver function and normal liver biopsies on after 5 days. And on the limbs, it was 7-day follow-up. And I am with you. If you do a very short follow-up, and I am with you on that, there's a lot of data on the literature that they do something with the organ, put it back for a few hours and tell, this is working great. I don't like it. So if this is your question, this is how I see it.
Other questions? Yeah. So I think as you want to, there are multiple systems in place, and you get great information on the genomics and proteomics and metabolics associated with your system. Is there a comparison between the different perfusion machines as far as following those cytokine/endpoints or genomics? Because I think it's great. I mean, obviously, the different systems and you all compete, so it's hard to have an outcome that's similar.
This is a great question. Thank you very much for this question. When I present this in Europe, the guys love it and want to send me all the tissue samples. It costs a ton of money. To do this costs around $100,000 for this thing. So I don't think that most of these people will generate this data because it's not required by the FDA. So you and I, as a surgeon, we might want to know what happened in metabolomics and genomics assays on this. We probably have to fund our own work for this because they're not going to do it for us.
So it's hard to compare which one's doing what relative to the other one.
Yes, but when I was in Europe last year at ESOT, they showed some liver data, and I saw some of the work that they did similar to this with the warm perfusion. It looks a little bit different as far as the inflammation itself and the level of some of the cytokines. But do we have a side-to-side straight group 1, 2, and 3, the same variables? We don't. But you're right. We should have it. You're absolutely right. a great question. You're very smart. Yes, the issue of trying to minimize the ischemia reperfusion damage by giving early oxygenation is on the table, but legally could be a problem to run a clinical trial, because if you're going to do this, the insurance company will say this is not standard of care, and they will make you pay for everything from the time that you see the donor, and I didn't have the money for this. So your second question is, if this is true, can you oxygenate the donor right away? And that is even more complicated legally, because in Europe, they are asking my solution right now to do exactly what you said. You get a DCD, you're going to recover the organs, flush the blood out, and put this solution in, oxygenate the organs right away, take them off the field, drop the temperature, life is great. In our country, the lawyers said, you can't do this, because now you could oxygenate the brain again of a person who was legally dead, and you couldn't do it. So you are absolutely right that we should find a way to do this early on. Logistically, right now, with the money that I have, I can't do it, but you're right. It's a very good question. We were crazy about what happened with the clotting system. So I was trying to run TEGs early on. I didn't have the support to do this. We went to a PT, iron, and platelets. They look around the same. But when I started breaking down the data on the genes and on the metabolomics, it was clear that the liver was producing more clotting factors. Do we become hypercoagulable, which is a problem? When you put the liver back in, if it clots well and everything clots, we got a problem. So I did not see this. I don't know if you operate on pigs yet, but the pigs are a little bit more hypercoagulable than you will like, because this is how they are. So when I did this, I was really thinking of what you said. Are we going to celebrate this and come back the next day and everything is clotted? So we did 7 animals like this, and every single one at the necropsy, the vessels looked great, and there was no sign of hypercoagulation in any animal. But we don't have the data that you're asking, looking at the TGs and the clotting factors in an organized way. We don't have the data. It's a very interesting topic. When I got into this field, I met a group of my colleagues. Dr. Badalak is the leading on this, doing this for 10 years. And they have on and on papers showing that there's no DNA left. If you look at publications on large animals and on rodents, there's no DNA left that is viable cell on this. The problem with this 3D scaffolds is the following. If you start infusing cells on, how can you oxygenate and feed these cells? Because I did a lot of cell cultures on the flat place where you have the cells in the hyperbaric chamber on, then you have oxygenation to the flat cells on. Or I also culture cells. I did islet culture on the matrigel, and you have a few layers of cells growing. When you have a 3D system like this, and they were seeding the cells back in, the cells die within a few days because of lack of oxygenation. So it's very interesting that when you try to reconstruct, when you try to rebuild these organs, and this is why I fought with these guys in a nice way. I said, they want to just put some cells in and transplant the organs back. I said, you got to be kidding. You got to have a bioreactor to grow the cells back in. This is what we're doing right now. This is very labor intensive.
Thank you very much.
Thank you. Thank you.